So this is our DNA structure, and it's a double helix shape, as you can see. And the DNA nucleotides are consisted of one deoxyribose sugar, which is shown by the black chocolate munchkin, and this is a phosphate, and then it has all the bases on it. So there are 4 kinds of bases: adenosine, which is the red; guanine, which is like orange; thymine, which is a green. Thymine, which is the yellow, and then cytosine, which is the green. So the adenosine and the guanine, they're called the purine bases, and that means that they have double carbon rings. And then the thymine and the cytosines are called pyrimides, so they have only single carbon ring. The complementary basis for adenosine is the thymine, and they're connected by 2 hydrogen bonds. And guanine and the cytosine, they're connected by 3 hydrogen bonds. So this is a hydrogen bond that are formed between the bases. And I'll now talk about the replication process. So to break the hydrogen bonds, an enzyme called helicase, which is a goldfish, comes to break up the double helix. So here it's already broken off. And then
this is the leading strand.
Right. So I'm going to talk about the leading strand, but yeah. So as the helicase breaks apart the DNA, this is 5' and 3'. So the DNA polymerase 3 comes following the helicase and breaks up, follows the helicase, and then it forms the complementary base that matches the original strand. So in this case, it's adenosine, so it's gonna make the thymine and form hydrogen bonds first. Okay, I'll just say. And then the phosphate, I'll just do everything. Okay, and then the phosphate and the sugar, they're combined, they're connected by covalent bonds. So they just go straight up and then form hydrogen bonds.
So that's the leading strand, and since it goes from 5' to 3' on this way, which is not in the direction of the helicase, this is called the lagging strand. So what first happens is that an RNA primase is alongside the helicase, and so they travel in the same direction. So as it's traveling across the lagging strand, it forms RNA primers, and these RNA primers are sort of like an SOS signal. So that tells the DNA polymerase 3 to come along and make complementary DNAs, which again go in the process of a hydrogen bond and a covalent bond, another hydrogen bond, another covalent bond from, again, 5' to 3'. And these, since they're separated by the RNA primer, these are called Okazaki fragments. And then what happens next is that the DNA polymerase 1 comes and changes these RNA primers to complementary DNA strands. And since they still are separated, an enzyme called ligase comes along, which is basically a glue and glues these fragments together. And this is demonstrated with frosting. So, yeah, that's how DNA is replicated.